Okay, the last talk, and certainly not the least, is from Kartik. Kartik's assistant professor in the Department of Medicine, and his primary is appointment in pulmonary crit care. And I'm here on behalf of Lance Torrada to introduce him. But he also has a joint appointment in our center, and his labs are actually on Y6. A little bit about Kartik's background. He grew up around Houston, went to UT Austin as undergrad, and then did MD PhD here with Ralph DeBardinas. Then went to New York City for his pulmonary crit care training, and he was in New York City's ICUs during the height of the pandemic. So I've always been impressed by that. He then went on to do some postdoctoral work with Jeffrey Friedman, very famous guy. And Kartik has uncovered, I think, a really interesting and novel area of leptin biology. And I won't steal his thunder, but I think Kartik's own lab, he's going to be interested in the intersection between metabolism, altered or disordered metabolism, and immune function. So Kartik, the floor is yours.
Thank you. So last one here. So I'm also the second of Ralph's trainees. Ashley had a great talk, and I bet Ralph is pretty nervous right now about whether this. I'm just kidding. It's probably fine. So thanks so much, Joel, for the introduction. My name is Kartik Rajagopalan. I direct a lab, as Joel said, that's in the Center for Hypothalamic Research. The other 20% of my time, I'm in the ICU, the medical ICU, where I attend at Parkland Hospital. And I'm going to I changed the title of this talk. I called it Leptin Signaling Maintains Core Temperature During Severe Influenza Infection. It is different from the flyer, so that's on me. I don't let it confuse you. So, if you sort of saw me in the hall and asked me, like, what is your lab doing? We were just in private. I'd probably tell you, come back in a few years, and I'll give you an answer at that point. But obviously, if you're holding me to like something and what exactly we do, I'd say we work at the interface of infection, metabolism, and the brain. So what do I mean by that? What I mean is that right now, as you guys are sitting here in this room, you're probably running some kind of a basal metabolic program. You are active, you get hungry, you eat, satiate that hunger, and you have no trouble regulating your body temperature in a very tight range. However, when you get infected, this all really changes. You get really tired. You might feel like you don't want to eat much. Food might taste differently, and your temperature will start fluctuating. So you can become hyperthermic and develop a fever, or alternatively, some people will get hypothermic and actually have lower than normal temperatures. And why is this all happening? I think it's because your body is trying to reorganize its energy expenditure. So what I'm going to call your healthy energy expenditure that you normally sort of use is going to ramp downwards. And the reason it's going to ramp downwards is because I think the sickness-associated energy expenditure is going to have to ramp upwards. And by sickness-associated energy expenditure, what I mean is the energy expenditure that's required to actually for cell proliferation requires a lot of ATP, a lot of macromolecules in order to fight an invading pathogen very quickly before it takes over your body and kills you. And what do I think is the organ that coordinates all of this? I think it's probably the brain. So if you had to ask me, I'd say, what our lab studies are, how does infection change metabolism, and how is this metabolic change coordinated by the brain? So as Joel said, I'm an ICU doctor. So this is a very typical case I get in the ICU. So Mr. J is a 31-year-old man who has fevers, chills, and difficulty breathing. He decides to go to the hospital. In the emergency department, the nurse notices that his oxygen is 85% on room air. He is flu A positive. He's given supplemental oxygen, Tamiflu or oseltamivir, and admitted. While on the floor, his oxygen saturation falls further, and he's placed on high-flow nasal cannula. You guys probably remember during the pandemic, high-flow nasal cannula was a way to give tons of oxygen, about 50 to 60 liters per minute, in order to keep people's oxygen up. His body temperature then drops from 36.5 degrees to 33 degrees, and he requires a bear hugger or a heating blanket, and he's admitted to the ICU. So this is just a heating blanket that blows hot air. You can put it on top and warm the patient up. So hypothermia. So it's not that a majority of patients are hypothermic by any means, but it's not uncommon. So this is actually a analysis of 6,542 patients that were septic. And if you take a look, unlike our body temperatures, which are maintained mostly from 36 to 37 degrees Celsius, It's actually kind of a bimodal distribution. So you can see that some of the people are sort of have low body temperatures and some of them have fevers. And if you strictly define hypothermia as being 35 degrees, you see that about 15 to 20% of people actually fall into that range who are septic. So it's not an uncommon thing. But although it's not super common, it is a really poor prognostic sign. So this was a meta-analysis of 17,000 patients with pneumonia. And if you take a look at the way they presented and look at their odds of mortality, hypothermia is actually a 5-fold increase in mortality versus a fever, which is only 0.8. So that means that if you have a low body temperature, your association of dying, that was associated with a 5-fold increase of dying. So these people do very, very poorly. So I told you that it is not super uncommon. These people die more. Do we really know how to treat it? Not really. What we use is the same kind of thing that I talked about in the clinical vignette. We bring people into the ICU. We put them on what's called the bear hugger, which is a warming blanket. Now that would make a lot of sense to me if you were found outside somewhere in exposure in the cold, then you'd want to rewarm that person. But in this case, the body is doing it itself to the person. So the question is that really the right thing? There's no actual data to support it. And part of that is because we don't know the pathophysiology. So it remains debated whether this hypothermia is adaptive or maladaptive in sepsis. And if it is something that's adaptive, then what are we doing exactly rewarming it? So we wanted to study some of this, and a mouse model is easy because there are a lot of genetic tools available and they get a lot of the infections that humans get. And so what we do in the lab is we basically infect mice, and this is sort of a video that I hope will work. And we infect mice, and we basically put them under isoflurane anesthesia. We hang them by their incisors, which are surprisingly strong, on this rack, and turn them around. And then we put isoflurane is going through a little nose cone, so they stay asleep. And then somebody actually just, you can see me or someone else dripping influenza into their nose. And the nice thing about this is it's a quick infection, and they wake up within it takes about 2 to 3 minutes to infect a mouse, and they wake up within about a minute or so. So pretty quick. And they have a lot of the same signs that humans have when they get infected. You can see here that their food intake drops and plummets to about nothing at about 6 days or so. Coordinate with that, their body weight also drops a ton. So they basically end up natering about 8 days or so. And if you look 4 days later, some of our influenza viruses have a luciferase reporter on them, so you can actually see the virus and where it is. You look 4 days later and you see that it's sitting in the lung. So it does make an influenza pneumonia. And along with the pneumonia, you can see that we can measure the actual oxygen saturation. The pneumonia causes lung damage. So this is a mouse with a little pulse oximeter on it. They don't really like it that much, obviously, but it'll measure their oxygen saturation. If you take a look, these blue dots represent what it would be like if you just gave the mouse saline in its nose. There's no change. Whereas flu, over time, as it proliferates and does more lung damage, you start getting oxygen saturations that go down to about 60 to 70% or so. And then what about core temperature? What about core temperature? So they also maintain before infection at pretty tight range, maybe a little bit higher than ours. But as you can see, as they sort of end up getting infected and the influenza infection gets worse, they also go down to about 32 degrees or so. So they get hypothermic as well. And I basically took these like 10 mice or so that was in this group and plotted them here. And if you look and see the mice that are in the red are the ones that die and the ones that don't survive. And so if you look about at day 7 or so, these are actually the ones that are actually the most hypothermic as well. So there seems to be some sort of way to say, okay, well, this mouse is really low on this day. It's going to die. Again, don't know if that's causative at all. No idea, but it does seem to be associated similar with sort of poor outcomes in human patients. And so I'm just going to tell you about one way we're trying to interrogate this. So leptin, as Frankie sort of introduced earlier, it's a protein hormone. that's released by adipose tissue. It was cloned by Jeff Friedman and colleagues in 94. It goes and it has a ton of receptors. The most best known are in the hypothalamus, but it has receptors all over the place. Some of these receptors have been characterized actually by Joel Elmquist and colleagues. And so it binds leptin receptors in the hypothalamus and actually manages sort of sympathetic output. So things like temperature, heart rate, blood pressure are all known to sort of be regulated by leptin through sort of these receptors. And so we're going to focus quickly on temperature. What we did was we developed a model of leptin deficient OB mice. So these are mice that have a spontaneous mutation in leptin, so they don't produce any. And we can either put a pump that infuses leptin at 75 nanograms per hour or saline into the mouse 1 week prior to infection and then infect the mouse. And then the leptin pump or saline pump stays in for 2 weeks. And so what happens? Are we able to change anything with temperature? And the answer is yes. So if we essentially look at the core body temperature on various days, the mice that are in black are the ones that only get the saline pump. So these mice have no leptin throughout infection. They end up really tanking and dying, and they basically get very hypothermic. And also the mice that are in the red are the ones that maintain their temperature. And also survival also is sort of associated with that. So you end up having 100% survival in the mice that get the leptin, whereas the ones that end up getting the saline and have no leptin end up having about 80% mortality or so. So what I told you just now is leptin signaling the brain is responsible for maintaining core temperature during influenza infection. We're showing that in a mouse model. We're still trying to figure out what is the mechanism by which this happens. So there are multiple leptin receptors. And I think that by using a lot of the tools that Joel and the Hypothalamic Center have, we can sort of knock out leptin receptors in different tissues to figure out what is the mechanism by which this happens. Additionally, that could sort of also give us other therapeutic targets. And then the other question that I sort of talked about earlier is the hypothermia. Is it causative? Is that why they're dying? Or is that sort of just an associated phenomenon? It has nothing to do with it. And I think we're trying to figure that out as well. So really quickly back to our patient, his oxygen saturation drops further, requiring intubation. Now his blood pressure drops from 130s over 70s to 80s over 60s, and he's placed on Levophed or norepinephrine. This happens a lot in the ICU. And what I wanted to highlight here is that it's interesting, but a lot of the actual medications we give to stabilize blood pressure, they're all sympathetic neurotransmitters. So dopamine, epinephrine, Levophed. So my thought is that there must be some connection in how the brain sort of works during infection to sort of maintain these autonomic functions. And I want to sort of, one of the big goals of the lab broader is to figure out what happens and why this may not work or how we can exploit these pathways therapeutically during severe infection. We have these autonomic instabilities. So that's all. Thanks a lot. My lab is small. Toddy's a technician's been fantastic. Roger now started co-mentoring a pulmonary critical care fellow with Joel. A lot of other people, and it's been great to come back here to UT Southwestern, and I'll take questions. Thank you.